Now that we've explored the techniques, let's look at some more applications of biotechnology. By examining a person's DNA sequence and identifying mutations, several genetic conditions such as sickle cell disease or Huntington's disease and haemophilia can be diagnosed, or disease risk can be assessed. We saw how cloning produces insulin, and it can also produce other human proteins such as clotting factors by adding the gene to the genome of mammals. So here, a transgenic goat secretes thrombin in her milk. Human papillomavirus is associated with cancer of the cervix in women. To create a vaccine, Professor Ian Fraser and his team at the University of Queensland isolated the HPV capsid protein gene and introduced it into a vector. When the recombinant vector was inserted into the host cell genome, it produced empty virus-like particles which were then used as vaccine products against HPV in humans. Transgenic crops have three main aims: pest control by inserting a gene that protects plants from certain pests, and this was previously undertaken by spraying with the pesticide. Weed control by inserting a gene to protect against herbicides so that weeds die when sprayed but the crop isn't affected. And then crop improvements by inserting a gene for a vitamin or resistance to drought, or knocking out a gene that produces a human toxin. As mentioned earlier, an example, or a great example, is golden rice, developed to express genes that produce vitamin A. Vitamin A deficiency is the cause of blindness and a big problem in many developing countries. Let's reflect on the learning objectives we've covered. You should now be able to describe how DNA technology can have medical applications in such areas as the development of gene therapy, vaccine production, and the development of pharmaceutical products. And describe how gene manipulation has practical applications for environmental and agricultural work. In 1997, Scottish researchers announced the birth of Dolly, a lamb cloned from an adult sheep by nuclear transplantation of a differentiated cell into an enucleated egg. This is termed reproductive cloning, the production of a new individual. This image of Dolly was an image that I took when I saw it at the National Museum of Scotland in Edinburgh. It's important to note the difference between gene cloning and cloning of an organism. The process is inefficient, as few develop to birth and many cloned animals have health issues or defects. Dolly, at the age of 6, was found to have tumours in her lungs and was euthanised. This type of thing is usually seen in much older sheep. The early ageing was believed to be because differentiated cells have adult epigenetic patterns which interfere with the gene expression patterns and process needed for embryonic development. Dolly was the first reported case of a mammal cloned using the nucleus of a differentiated cell. The researchers initially obtained mammary cells from a donor sheep and cultured them to cause de-differentiation. The arrested cells were then fused with enucleated sheep eggs. The resulting diploid cells divided to form early embryos, which were then transplanted into surrogate mothers. Out of several hundred embryos, only one successfully completed normal embryonic development, and Dolly was born. Analysis proved that Dolly's chromosomal DNA was identical to the DNA of the mammary cell donor sheep and not the egg cell donor, and you can see that clearly here with Dolly and her surrogate mother. Since 1997, cloning has been demonstrated in many animals, or many mammals, including cats, cows, horses and dogs. And in 2016, it was reported that Dolly's siblings from the same cell line are in good health. Although, for obvious reasons, human cloning is banned, this type of cloning technique can produce a range of differentiated cell types from an undifferentiated stem cell. This is termed therapeutic cloning. Embryonic stem cells are pluripotent, capable of differentiating into many different cell types. However, this technique is controversial because to obtain embryonic stem cells, an embryo must be destroyed. Adult stem cells are multipotent and can differentiate into some, but not all cell types, and they're also difficult to culture. With new techniques, researchers have been able to produce induced pluripotent stem cells from differentiated somatic cells. In this technique, skin fibroblast cells were obtained from a patient. Using a viral vector, 4 stem cell master genes were introduced into the cells. As the stem cell master genes were expressed, the skin cells were reprogrammed into becoming pluripotent stem cells. The pluripotent stem cells were then treated with specific growth factors and transcription factors to allow differentiation into specific cell types. These cells could then be returned to the patient to repair the damaged tissue. Early concerns about the potential danger of recombinant DNA technology focused on the possibility that it might lead to a deliberate or accidental release of new bacteria or viruses with increased pathogenicity. To prevent any such incidents, a set of strict guidelines have been developed that are followed by many countries. These guidelines are to have containment laboratories and strict laboratory procedures to prevent microbes from leaving the laboratory. In addition, the bacterial strain to be used in recombinant DNA experiments should have low viability. And finally, regulations are in place to control and even ban dangerous experiments. Despite the clear benefits of genetic engineering, there are a number of concerns about this type of technology. The majority of GM organisms, or genetically modified organisms, are plants, and concerns have been raised about environmental and health impacts. However, the approval and testing process is extensive, and in Australia involves the Office of Gene Technology Registrar and Food Standards Australia and New Zealand. Foods derived from genetically modified crops have been consumed by hundreds of millions of people globally for over 15 years. Hundreds of studies show their safety, including many independent studies. Numerous strategies are used to manage environmental concerns, such as rotating crops and adding genes to cytoplasmic organelles so that they're not inherited. Let's reflect on learning objectives now. You should be able to describe reproductive and therapeutic cloning and know similarities and differences and why these techniques may be controversial. You should be able to discuss some safety and other ethical questions related to recombinant DNA technology and studies and the biotechnology industry. Now that we can use techniques to sequence and analyse the genome, it's been revealed that 98.5% of the human genome does not code for proteins or RNAs. Surprisingly, it's been found that there is little difference in the number of genes between, for example, worms and humans. It's believed that much of the complexity in humans comes from sequence rearrangements, alternate splicing, and post-translation modifications. There are 3 main classes of DNA sequences in the human genome. Only about 1.5% of the total human genome codes for proteins or is transcribed into rRNAs or tRNAs. The introns and regulatory sequences that promote gene expression make up about 1/4 of the genome. A significant portion of the genome is repetitive DNA, and about 15% of the genome is unique non-coding DNA. Up to 50% of the genes in the human genome are single copy genes. Duplicated genes are 2 or more close copies of a gene which occur due to an evolutionary event such as a chromosome rearrangement. These genes form families that encode protein families such as the globin gene family, where 2 related gene families encode for the alpha and beta globin chains, and these are the polypeptide subunits of hemoglobin. Recall we looked at sickle cell disease case study that affects where a mutation affects the beta globin chain. The gene families also contain several pseudogenes labelled in gold here, and they no longer encode for any functional proteins. Pseudogenes are sequences that have accumulated mutations and therefore have become non-functional. Both eukaryotes and prokaryotes have stretches of DNA that can jump from one location to another, and these are called transposable elements. This activity can disrupt genes or facilitate recombination between non-homologous chromosomes. Hence, they play a role in genome evolution and potentially disease. Eukaryote transposable elements are of 2 types. The first types are transposons, which move within the genome using a DNA intermediate, and the others are retrotransposons, which move using an RNA intermediate. Transposable elements are the most abundant type of repetitive DNA sequence in eukaryotes. Satellite DNA, repeated units from 1 to 500 base pairs, is thought to have occurred due to a mistake in DNA replication. The number of copies of the repeating unit can vary from site to site within a genome, and most are located at the telomeres and centromeres. There are different subclasses of satellite DNA based on the length of the repeats. When the repeat units have 2 to 5 nucleotides, they're called a short tandem repeat, or STR. Similar to any other genes, satellite DNA can have various alleles, so they can be polymorphic amongst individuals for the number of repeats at a location. Only identical twins have the same number of repeat sequences throughout their genome. Satellite DNA can be useful information for DNA fingerprinting or forensics and paternity testing applications. DNA technology is widely used in forensic science and paternity testing. It uses the variations in the length of these short tandem repeats, or STRs. These are these randomly repeated units of 2 to 5 nucleotide sequences located in specific regions of the genome. The number of repeats are highly variable from person to person. So we're not going to go into the detail, but for interest's sake, let's just have a look at how this works. As shown in the figure, 3 individuals have a different number of repeats at this particular STR locus. Usually 10 or more STR loci are examined to provide greater discrimination between individuals to help with identification. This information can be used to identify people. So using PCR, the STR sequences are amplified and the length of the region, and thus the number of repeats, is analysed. It represents a particular allele. And then, of course, it's determined, the length of the sequence or the region is determined using electrophoresis. The paternity of a child can be determined as a child inherits alleles, of course, from both parents. So in example 1, we see a match as the child's allele matches with the allele from the father. However, in example 2, the allele of the child doesn't match with either alleles from the father. So this would be considered a negative paternity result. This type of technology can also be used in constructing family trees. For example, how's this one? Using DNA samples from mummies, researchers were able to extract repeating sequence data from which they have constructed a family tree showing ancestry in King Tutankhamun's family. Note the first degree consanguinity here in his parents, who are shown to be brother and sister. And as we know, King Tutankhamun had several congenital abnormalities. So let's reflect on the learning objectives we've covered. You should be able to describe the different types of DNA sequence arrangements, explain how transposable elements can contribute to genome evolution, and explain how repetitive DNA sequences are useful for forensic science and to be able to determine paternity. And of course, you don't need to go into great detail, but just a broad overview. Thank you for watching.